Hi, I'm Ted Johnson with QIAGEN Digital Insights, and this is Courtside View. This is our weekly vlog where we talk about data science and topics related to managing data and biology. I have with me here Allison, who started her biocuration career with Ingenuity Systems and became part of the QIAGEN family when Ingenuity was acquired in 2013. Throughout the past almost 25 years, she's had some involvement with all of the manual curation projects here at QIAGEN. And we thought it would be great to have a conversation today about how we do curation and why we think it's important. Allison, welcome.
Thank you.
All right. I thought we might start with the question of, why might manual curation be a better option than AI methods, for literature review in this age, where there are so many options to bring in, AI agents and AI models.
So when we think about literature curation and literature review, we're often talking about very complex biological systems and nuanced information. And then on the other side, when we think about our customers and how they're using this information, these are very important things that they're doing. It's things like biomedical research, it's drug discovery, it's variant classification for patients with diseases. So we want to make sure that our customers receive the most accurate and precise information. So while I think that AI methods definitely have some pros, they're very fast, they're scalable in a way that human curation is not, I think we ought to be aware of the limitations and make sure that we're accounting for those when we include information. So some of those limitations might be that we don't know necessarily what an AI has been trained on. And they only know what they've seen. So they may be including low quality data, maybe non-peer-reviewed information, possibly meeting abstracts that turn out to be non-significant information, but it's been included. So because AI kind of just takes everything and gives it equal weighting, that curation can tend to be noisy. So we want to make sure that we're checking it and curating the accurate information that is well supported in an article. Nuances in language not necessarily handled very well by AI in the way that a person can understand things. So in that respect, I think literature review by AI is very similar to everywhere else that we use AI, that it's a nice tool that we can use, but we need to check what we're getting back to make sure that it's accurate.
Yeah. So, I really take it to mean, when we think about these AI agents and these AI tools, they can't apply that same level of judgment that maybe a human expert could.
Yeah.
And that's what we're supplying here is that level of human reasoning and human expertise that
Exactly.
models can't hold. And
Yep.
when we talk about manual curation, what do we mean when we say that? I know at QIAGEN, I say manual curation all the time.
Right. So for us, it means that a person has actually checked over that piece of information and made sure that it's an accurate representation of what's in the original document. So when we're talking about manual curation, it's not just journal articles. We also manually curate drug labels, treatment guidelines, clinical trial records. Each of these types of documents, they have a different format. And so a person will look at those different types of documents and extract the information that is necessary to power our applications. We don't do any kind of inference or interpretation of the data that we see in the documents. We just capture as stated by the authors. This is important because we have a central repository of the data that's been curated, but that central repository powers all of the QIAGEN applications. So if you're curating, say, a drug approval finding from the FDA, that finding may end up in Ingenuity Pathways Analysis, but it can also end up in QCI for variant interpretation. So we want to make sure that we capture the core piece of information, and then our applications will use it as intended by the different applications have different intended uses. So when we're doing manual curation, we're also looking at the entirety of the document. So that includes the published document, like if it's a research article, it might be the actual document that appeared in the journal, including tables, including any figures, but also including supplementary information. So oftentimes, if Say if you're curating a PubMed article, you may be able to access the full text of the PubMed article in PubMed, but the supplementary data is on the journal's website. So they're not those pieces of information that are related to each other and together comprise the entire article. They're actually separate places. And in manual curation, we are also looking at that supplementary data and bringing it into the findings. So I had a couple of examples of curated findings that I could share that demonstrate this idea of how we synthesize a finding. They're very simple examples, so obviously sometimes it's more complicated than this. So in the first example, this is an IPA finding, Ingenuity Pathway Analyis. So the finding as it appears in the application is shown in the middle there. It's a very simple finding about the effect of microRNA on proliferation of endothelial cells. This finding would have originated from an abstract sentence. So if we look in the abstract, we see a sentence that miR-150 changes the proliferation of endothelial cells. So a curator looking at this piece of information in the abstract would then ask themselves, well, what kind of microRNA is it? Is it a 5P? Is it a 3P? Is it the precursor form? Is it from human? Is it from mouse? Is there a way that I can tell from the article, other areas of the article, what was actually used? And then similarly for the endothelial cells, what kind of endothelial cells? Were more than one type of endothelial cells tested? And is there a way that I can see that? Was the same effect seen in all of those cells that were tested? Did they come from a human or did they come from a mouse or some other organism? Is it an in vitro model or was the test actually done in an animal model? And how was the assay done? So what kind of method was used? So all of that information will be gathered from wherever it is in the article, if it's available, and put together into the finding that you actually see in the application. So the second example I have is from QCI. So QCI is a variant interpretation software that we have. And again, you can see the finding in the middle of the screen. There's actually two findings here. One is for the presence of the variant in a patient that has a phenotype, and one is for looking in a population of healthy controls for the variant. So in this case, the variant is mentioned in a table. It's actually found in 2 patients. We can look in the materials and methods, and we can determine what the ethnicity of the patients were. What is the age of onset? How was this testing done? In the results section, we can see mention of a functional assessment, and there would be data in a figure that supported that. And then we also know about the population of controls that it was looked for. So a person that's using QCI can come here and see this reference, and which is a link to the original PubMed article, and then they can go out to the original article and see the information as well. All of the findings that we capture by manual curation are traceable to the original source. So this variant was actually found in 2 patients, and that is not represented in this natural language version of the finding, but it's actually present elsewhere in the application. So if we go to on the variant details page, we have this section that represents the number of patients where the variant has been identified. And we can see here, we call it a manogram, the little icons that represent the patients. If you click on one of them, actually 2 of the icons light up, and that shows us that these 2 people were found in the same article, and it will be linked back to that reference. So the manual curation process is responsible for synthesizing all of that information and capture it in ways that the applications can use.
Wow. So what I take away from this is that, as a human expert, you're able to synthesize, data from multiple parts of the paper to really understand what the authors were trying to do.
Yeah.
And the other thing I often highlight when I'm talking to people about training models is it's a probabilistic model that's trying to emulate what a human expert would do given the same data. And really, I'd say that you're setting the gold standard for what human experts can do when given this biological data.
Absolutely. And it's not that there's no place for AI, because I think it's a great tool that we can use for identifying articles, maybe extracting the AI version of findings. But I think ultimately it's best if a human is involved at the end to make sure that we have the most accurate data, get rid of conflicts of interest that might be present that wouldn't be detected, any kind of bias in the information. I think they can definitely work together.
Absolutely. I think, bias is so hidden
Yeah.
by its nature, but also for AI agents. They can only know, what was in their training data. And, I think we've seen they can't parse humor necessarily, and they can't parse, uh, quality the same way, an expert could.
Yeah. You'd actually be surprised how often there's typos in scientific articles too, in gene names or in variant descriptions, and it really throws you off. You have to discern what was intended.
Absolutely. And I think that's a good place to segue into, who does this menu curation for QIAGEN?
So we have a group of curators. We have over 100 scientists. They are PhD or medical doctors, MDs. For each of our curation projects, I already mentioned that we have different types of documents that we're curating. For the different types of articles, we have a curation project. That would be it would have a protocol associated with it and possibly something we call a curation tool, which is like a glorified form that you fill in while you're doing curation. So each of those curators has been trained on the particular protocol and tool that is relevant to the project they're working on. And we also have a competency assessment. So during the training process, we do the training, we assess competency, and that's an ongoing thing where we're constantly assessing the quality of our curation by each of those curators.
I think that's excellent because one of the things I know from, the training of these large language models, we tend to harvest lots of information from the internet. But that means that sometimes in that training data, what you're getting, your answers from is Reddit or Quora or, blogs from, uncontrolled people. And it's important, I think, to know who produced the data, who is working on this, and are they an expert in their field? That layer of judgment, I think, is significant and something that, can't be done artificially.
Absolutely. And while we're not inferring or interpreting data, we do use our scientific judgment to say, what variant did they intend to say here where they've put a typo in the description, or, how does this fit in scientifically?
Absolutely. And I know we've touched on a few. I know we talked about QCI and we talked a little bit about Ingenuity Pathway Analysis, but what all are we doing here at QIAGEN with this manually curated data? What kind of applications might our customers see?
So the manually curated data is present in the knowledge base, and that knowledge base powers all of our applications, with the exception of BKB.AI, which is all AI. But all of our other applications contain manually curated information. So on this slide, you can see I have a number of different types of sources highlighted with arrows going to the different QCI applications, and almost everything is in almost everything. Omicsoft is slightly different, but everything else is there. So all of our applications are seeing manually curated data.
Wow, that's a powerful way to access, that human expertise.
Absolutely.
And, on the subject of expertise, how do we maintain quality? I mean, it's a big lift to go through all of this documentation.
Exactly. So as I mentioned, we have training and competency assessment of our curators. So you can think of our path from curation to the application. It's a linear path in this case. So we have the curation, we hire our curators, we train them, we make sure that they're competent to do the curation, and then they start curating. And so they're curating typically in online curation tools. These tools are kind of forms, so they have slots that are looking for specific information. And then we have curation protocols, which describe to the curators where to find the information in the document, what kind of information they're looking for, and how it should be captured. And then we also have built-in constraints within these tools. So these are little tests to make sure that people are putting the right information into the slots. So for example, one constraint might be that you can't put a cDNA level mutation description on a protein. That would be a constraint. And if you tried to do that in the tool, you would get an error message and it would tell you how to correct it. So after the curation process and the curators are finished curating, they submit their work. And then we have a a spot checking process in place. So this is a second person that will go and look at the document or the curation of a document and check to see how it's been curated and make sure that it has followed all the rules and everything looks good. So after curation, the findings are uploaded into our knowledge base. It's a central repository. And then within the knowledge base, we have a number of other constraints. And this is checking that all of the information within the knowledge base is consistent. And so we have constraint scans. They're run on a nightly basis, and they will send warnings if any kind of issues are identified, and then we can go into the knowledge base and fix them. When it's time to do a release, we do a build process, and some of the files that are generated from that build go through a post-processing process, and then they're uploaded into kind of a staging environment before release. And at that point, we have end user testing. And this is checking to make sure that the data that we've curated is appearing properly in the application. In the case of QCI, we have ACMG and AMP rules that are used to classify variants, and we're checking to make sure that the rules are being triggered appropriately based on what has been curated. And then finally, the information goes into the application, and we await, and if there's a problem with it, we will hear from customers, but typically we're pretty good.
All right.
Cut out that last bit.
Yeah. I mean, many of our customers are themselves scientific experts, and yeah, they're definitely experts in their field. So what do those users do if they feel like they've identified a gap in our knowledge base?
That does happen. So if a customer identifies a gap, they can contact customer support at the tsbioinformatics@QIAGEN.com email address, or their FAS or salesperson that they've worked with, and those people can pass along the request to the curation team, and we'll assess whatever is being pointed out to us for inclusion.
Yeah. And I think this involvement of our end users and the scientists who use our products, that's in some ways another strength, I would say, of that curation process, is that you have that back and forth with the experts in the field.
Absolutely, because we're not experts in everything. We know science, but not specific areas or topics particularly. So it's really helpful to us when customers can point out to us what might be missing. So it can only help.
Absolutely. And I know, as a person who does a lot with computer science, sometimes AI can't handle the, complexity of the biological field, that, biology is squishy, and or even the terminology around it can be a challenge. So what are some examples of where AI can't cope with that complexity and where manual curation really shines?
Right. So in science, well, in biology, we have this concept of a genome build, for example. So a genome build is like a reference for all of the DNA contained in an organism, and it's organized by chromosomes. And the way that these genome builds are put together is that we sequence multiple organisms of a particular type. We gather the information, and then we put it together and say, this is what is representative of a typical organism of that nature. Now, sequencing technology is always evolving. So as it evolves and we get more data, we can fill in gaps that we might have. We can make corrections. And so when a next genome build is released, the positions of genes have changed. Sometimes the names of genes have changed. In science, you might have multiple groups that identify a gene at the same time. They all give it a different name. And so we need some kind of normalization process to say, oh, that gene is the same as this other gene, and actually it maps to this. So there is a standard list of gene names, but there's a lot of synonyms around as well. In addition to that, each gene may have multiple transcripts, and only some of them may be clinically relevant, but other ones are available too. So all of that information needs to be put together, and even when we do curation, you have to keep in mind, when was this article released? What genome build were they looking at? Do we need to do this liftover process to move it further along to the next genome build? So AI has trouble with those kind of concepts where it's so complex and everything is changing and you need to keep up to date. And it's not always obvious what models or what builds AI was trained on. So it's hard to say. And variants can be misaligned, genes can be misaligned, things like that. There's nuances in language. I think I mentioned that before, where like things like negation doesn't get handled well, speculative statements. So oftentimes, especially in abstracts, if you're looking at abstracts, towards the end of the abstracts, authors are speculating on what the implications of their research might be, but AI can interpret that as a factual statement that isn't supported by the article, and then it integrates that into whatever it's doing, and that can cause problems too. A lot of biology involves feedback loops, so it's not always linear. Sometimes, this protein, once it gets to a certain level in the cell, it feeds back and turns off expression of itself. And that is kind of a hard concept for AI to handle. A lot of the times, the amount of information available on a topic is not consistent. So we have areas of science where, this has been around for a long time. We have a really good understanding of how things work here, what the mechanisms are. So, new research articles in that area are just integrated into this big, large body of knowledge, and AI can handle that pretty well. When you have new concepts or novel mechanisms coming along, you initially get kind of a lot of information. Well, you might not have very much information at all, and that can be an issue. Or you may be having conflicting information coming in. So, one of the examples I think of is, you think of the early days of the COVID-19 pandemic, and there was so much information being released. A lot of it turned out to be wrong. A lot of it needed to be taken into context, like how was that experiment done? What system were you looking at? AI only knows what it's seen already. So all that kind of novel information, it would struggle with generally.
All right. I mean, obviously curation, it's a very nuanced process that involves, a human making judgment calls based on their expertise in the field. Where would our users go to ask questions about the curation process if we kind of inspired some deeper thoughts today?
Yeah, so they can always contact the bioinformatics customer support at ts-bioinformatics@QIAGEN.com, or any FAS or salesperson that they're used to working with. And if the person that they contact can't answer the question, it can be escalated to somebody in the curation team.
Awesome. To summarize what we've talked about today, we wanted to cover this concept of manual curation, and that's what we do here at QIAGEN. I mean, this is Allison's field of expertise, and her team really leads the charge in developing gold standard, data around the biological field. And we think that this is important, right? Because this data curation process really has, deeper interpretation or deeper reasoning involved in it than these AI agents are necessarily capable of. And these experts can develop that gold standard, that these models rely on, then to actually have factual data. And so, it's through this process that we really help inform our customers and build out our products. And, I'd say it's foundational to everything we do here at QIAGEN. Allison, it's been great having you to talk with us today, and we really appreciate you joining us.
Thank you so much for inviting me.
And, this has been Courtside View. Please join us again next month for our next discussion.
QIAGEN.
Sample to insight.